So there are scientific methods in biology, and those scientific methods are, like I said, they could be of different types. The scientific methods actually starts with raising questions. You're going to have a question, and that question needs to be answered. And in order to answer the questions, you look for data as information, you collect them, and you're not simply collecting a lot of junks, but rather you focus on those which are good potential answers, could be good potential answer source for your raised or formulated question. And after having those informations, you're going to come up with possible answer for the question you have already proposed. And these possible answers or scientific guesses are called hypotheses. It could be more than one, and again, you look for reasons. You can follow inductive reasoning or deductive reasoning, and using those different kinds of reasoning, you're going to come up with the strongest hypothesis, which we call prediction. So you have proposed question, you raise it or you have it, and you try to answer that question. You need to collect data, information, and there are not junks of every information what you come across, but could be a possible answer for the question. From this information, you're going to have hypotheses that are possible answers, and from these numbers of hypotheses, you're going to select the strongest hypothesis, which you call prediction. And the testing of this prediction, which is your possible answer, is what we call experiment. In the experiment, this prediction, this answer, whether it is the right or wrong answer, will be tested, and the outcome of the experiment is what we call result, and the result will be analyzed, interpreted, and then this interpretation, analysis of result is what we call the conclusion. In the conclusion, there will be a statement that falsifies the prediction, which is not the answer, or there will be in a conclusion a statement that verifies that the prediction is what was truly found as an answer to the question that you had, and this is going to be considered as a general theory after repeated experiment done by the very person who did it earlier, and by many personalities which are science personalities or scientists, and then it will be considered as theory. Again, that theory will, after a long period of time, through a number of observations, could be subjected to question. And again, that question needs an answer. Then you have a collection of data. And then from it you're going to have a hypothesis from which you may have a prediction, and that prediction is going to be tested in an experiment. So either there will be a very big loop that happens after a long period of time, or you may have a very short loop that is just go back to hypothesis after a falsifying statement by the conclusion. So the way it goes on is that question, data collection, hypothesis, and then prediction, experiment, result, conclusion, theory. This is one way. Another way is you have hypothesis, you have questions and data collection, and then hypothesis, and then prediction, and then the prediction will be tested by the experiment. From the experiment, you have a conclusion that falsifies the prediction, and back to hypothesis, prediction, experiment, result, conclusion. This is another loop. So one way or the other, we have a very big loop and a smaller loop in scientific methods. So there are different ways of the steps of scientific methods, specifically tailored to a situation raised. Sometimes you have an observation that brings about hypothesis. You observe something and then you try to explain the situation, and that's what we call the hypothesis. And from the different ways of explanations that you try to make, one will be stronger prediction, will be put into experiment, and then you have analysis of the experiment, interpretation of the experiment, and still that will be subjected to evaluation, and non-stop what we call cycle will be there. Biology, like I said earlier, has got a core tool and a core state. The core tool is what we call the microscope, and the core state is what we call the experiment state, where we have the testing of the prediction. Now you want to test the prediction and you're dealing with living things, and that really makes a little bit harder time, or gives a little bit harder time compared to physics and what we call chemistry, which are the sister sciences of biology. And here what we need is, we're going to have a control group of organisms and the actual experimental group of organisms. You're dealing with the experimental group of organisms in a way that you're going to test certain variables. Are you going to test the deficiency of vitamins? Are you going to test about the effect of certain food or certain mineral? Or are you going to test whatever you raise? You're going to test it on those experiments or experimental group. With this experimental group, in parallel, you must have a control group with which you're going to compare the result that you get from the experimental group. Everything between those two groups, experimental and control, is exactly the same. You treat them exactly the same, except for the variable. So you're going to see the effect of the variable as compared to what happens to the control group. For instance, because of the variable, if the experimental group are dead, and at the same time having the variable, the control group, are found dead or found with certain expected problems that we expect in the experimental group, then your result is not due to the factor, due to the variable. So they will tell us as to the result is towards the cause of the caused by the prediction. I mean, is that the prediction falsifying the result, or is that the prediction verifying result? It can tell us from the control group. So control group is there to let us evaluate as a touchstone of what we call the experimental group. The other thing that we may discuss about is the report writing. Whenever we have an experimental sessions, or whenever you have experiments, then there is a kind of formats. That's not exactly one single type of format. The format differ from kind of experiment to kind of experiment, but more or less the format may have or probably have almost all or some of those which we are going to discuss now. Actually, there must be always a date of the experiment. When you do the experiment, the data has to be specified. Then you're going to have the subtopic and the topics of the experiment. That will be followed by why the experiments, what is the objective of the experiment. Actually, you're trying to state there the first step of scientific methods, which is stating the question. Why are you doing this experiment will be one point, and then that will be followed by the apparatus. What are you using? You simply list them down. Droppers, hydrochloric acid, you have nitric acid, you have sample of egg, and all of that, just whatever you have, you list them down. Then you go to the procedures, the stepwise do's that you're supposed to do, and you list them, not an active voice, you're going to use a passive voice. You're not going to mention the subject of the action. That means you don't say, I did that, I do that, and you say, rather, it was done, it was placed, it was added, and so forth. That would be followed by the outcomes, the results. What did you see at every steps, okay? What color change you observe, okay? What events in the organism that you've seen, okay? What are the events that happen due to the experiment is what we are trying to put on the results. Then once you have the results, you're going to interpret the result. You're going to analyze the result. That's the interpretation. Again, this is going to be in passive voice. Conclusion, you're going to conclude after the interpretation of results, you're going to conclude whether what you suppose to be a solution, okay? Did you suppose the prediction is going to be an answer to the problem, or you proposed the prediction as an answer to the problem. Is it right? Is it a real answer, or is it an answer? Or if it's found out that it is not, it's falsified. So you conclude these things under the conclusion that will be developed into what we call the theory, or back to the hypothesis. So whenever you're reasoning in terms of inductive method and deductive method. In inductive method, you start out from general collected informations. In deductive method, you're going to support specific events. Okay. So in inductive method, it's the process of forming a generalization prediction from a number of observations. So from many, you're going to induct, get a specific statement. Or you have a specific statement and you support a specific statement. So this is deductive and inductive method or reasoning will help us deal with scientific study. Now in scientific methods, it's not always that you're going to get what you're supposed to get. That means whether to get your prediction right or whether to get your prediction is wrong. Sometimes off the line, you may come up with a certain and unthought and not in a plan and intentionally not existing, but very essential and beneficial. This time, that findings is not scientifically found. That finding is just accidentally found. Such an accidentally found, very landmarking, what we call discoveries of biology or whatever sciences, we call them serendipity. Serendipity is getting a result of an experiment whereby that result is very essential and a good answer to what you've not yet planned to answer. Like for example, the findings of penicillin. Penicillin is an antibiotic and it was found by accident. And this is really important. So it's not always not scientific, it's not always that through scientific methods that an achievement occurs in the world of science, but there are accidental achievements that are really beneficial to the human race. This will take us to microscope. I'll bring it. This is a very important tool, right? This is our axe in biology, with which biologists deal in life at the microscopic level and talk about the whole universe, talk about the whole atmosphere, talk about the whole earth. So microscope is very essential, unavoidably essential in the field of biology. So this is, for example, is one of the high school microscope. And there is a laboratory microscope, which is light microscope. This is a light microscope. We have also an electron microscope. And this electron microscope and light microscope, it will help us deal with the basic life process that's taking place within the cell. The crown characteristics of life, which we mentioned earlier, which is the metabolism. So this metabolism, as far as it is taking place inside the cell, it's quite unavoidable to study the cell, to know the fundamental principles of biology. So microscopes, this is one of the examples of microscope. Microscope has got the basic settings of lenses. For example, this is a light microscope. As I said earlier, there are two kinds of microscopes, the light microscope and electron microscope. Within the electron microscope there are divisions, and within the light microscope too there are divisions. We're going to just focus on the general divisions of the light and electron microscope. The light microscope has got two sets of lenses. One is closer to the object that we observe, and the other is closer to the eye. And this is what we call the eyepiece, and this is what we call the object, the three objectives that we have. So we have objectives, okay. I think we can see that. We have objectives. There are three objectives, a different power of magnification, and the eyepiece is there. Actually, the magnification goes to 1000 to 2000 if it is a light microscope, the maximum level that it may go, and its resolution, its separation between 2 points is about 0.2 micrometer, microscopic level of cells. For instance, we can see the egg of frogs, we can see even to some extent the egg of human being, and it will let us see some of the detail about those already viewed naked eye structures or cells to the level of the most small, the extremely smaller organisms, which are not, which we are not able to see through the naked eye. The light microscope can go on the magnification to 500 times, okay, from 100 times to about 500 times, a multiple of that of light microscope. And again, what we are supposed to see through the electron microscope is not necessarily being seen by the electron microscope, and the electron microscope being seen by the light microscope. But before dealing with microscope, it's better to go back again about the history of microscope. The history of microscope, Anton van Leeuwenhoek and Robert Hooke are most often mentioned people in the findings and discovery of microscope. Actually, the credit is given to Anton van Leeuwenhoek, who has been living from 1632 to 1723, though Robert Hooke also had his own microscope. Anton van Leeuwenhoek had a microscope, and he was the one who was able to see the moving unicellular organisms. In 1676, Anton van Leeuwenhoek invented a microscope, so it is given to him that the credit of invention. But the light microscope developed since then to the level of what we know today. Again, this development goes far beyond the light microscope into electron microscope, like I said earlier, and it was done by 1931, Ernest Ruska, 1926, Hans Bosch, who developed the first electromagnetic lens, and they are supposed to be credited for having the what we call first electron microscope. Electron microscope since 1915 started to be used widely. When we think about the electron microscope as compared to light microscope, electron microscope, a magnetic lens, and those has got light lenses. And electron microscope, in order to increase contrast between colors, you need, or contrast between, density different structures, you have to add metals. While in light microscope, you have to add some stains, specific stains. That will help you increase what we call contrast. The degree of resolution as compared to electron microscope, light microscope is shorter, while electron microscope is what we call longer. So when we focus on a light microscope, because light microscope is our high school topic, like I said earlier, there are two sets of lenses, eyepiece and objectives. Objectives because it is closer to object, eyepiece is because it is closer to the eye. This is the nose piece with which we are able to rotate. Okay, this is the nose piece, and this is the body tube. And it starts from here, all along the way is what we have the body tube. It has got an importance of preventing light strays inside. It allows the focal points, the crossing of the light rays properly coming towards the eye to harvest the lenses. And this one is a coarse adjustment knob. It will put the whole body tube up and down, and it helps us to find the image. This one is a fine adjustment knob. Okay, this is the coarse adjustment knob, the fine adjustment knob. We have the stage. Onto the stage we have the stage clips, and we have beneath the stage we have the controlling structure, the light condensing lenses that will bring about the cone of light, which is converting onto the object. And before it we have the controlling light amount controlling structure, which we have the iris diaphragm, and we have the mirror, which is serving as a light source. We have a light source here that will be reflected into the iris diaphragm, and the iris diaphragm will control the amount of light, and this amount of light will be formed into converging, formed by a condenser lens into a converging point where we have the object here, and that light pass through the object and enter into another lens, which is of a different power, medium power, high power and low power, and finally through the two lenses, okay, through the two lenses the image is magnified and seen through the naked eye. So the eyepiece, which is ocular lens, is 10 to 15 times. The objective, for example, our high school objective, 10, 40 times. Like I said, the multiplication of the objective and the eyepiece could go from 1000 to 2000 times. And we have other parts of the microscope with the eye point, the naked eye, through which we're going to see the image, with the body tube. That body tube allow the strays of light not to occur, forming the right focal point between the incoming rays, and forming the right focus or focal points of the incoming rays. The arm, which is a structure, is important to hold on. While I was talking earlier, I was holding this microscope by the arm and the base. We have a slide, and we have this slide, and the slide is placed onto the stage, and then we have the nosepiece, with which we are going to turn the objectives. Okay, we are going to turn the objectives, and that will be followed by condenser, which is forming a cone of light, beneath which we have the iris diaphragm. Then this sometimes is replaced by illuminator, which is a light source, and sometimes it reflects a light source into the diaphragm. This is what we call the base. We have the coarse adjustment knob to find out the image, and to clear and clarify the image is the fine adjustment knob. And sometimes this high school microscope can be used by the substage knob, adjusted in such a way that I will look through using such a position, or a position where I sit down and look. That would be simpler. And then if I want to look at the standing position, I can lower it down in such a way, like for instance, you have a seated, arranged object here, and you want the students to observe, then that will not be proper. This way will not be proper, because you want them to observe and go and observe and go. So this is the position which is good. But if you're supposed to allow them find out something while they sit down, you can give them in such a way that they're going to deal with the experiment. Now you have something onto the slide. Okay, you have something onto the slide, and this thing which is found onto the slide is objects, specimen. Actually, this lens is named after it, the objectives, like it was named after the eye closer to it, which is the eye point is here, and the lens is called eyepiece or ocular lens or ocular piece. So the slide differs from one kind of slide to another slide. You may have a permanent slide, which you're not supposed to change it. You don't have to prepare the object is already prepared. You just have to set the coarse adjustment knob and fine adjustment knob, and see what is there in the permanent slide. There's what we call the weight mount slide. It has got a stepwise process of doing the weight mount slide. There is a smear slide, which most of the time used in blood diagnosis in hospitals, in laboratories, that you smear the whole lens with the blood. Actually, in our level, in our high school, especially of grade 9, we only look at the water weight mount, what we call specimen, or preparing weight mount slide. The first step that you do is you put a drop of water onto the slide, having a given specimen, and then using a scalpel, you support the cover slip from either side and slowly allow the cover slip onto the object or the specimen, so that air bubbles won't be trapped, because the air bubbles will be creating problem while viewing under microscope. And the third step is, if the object needs staining, applying the stain from one side using a dropper. If it doesn't need, then you don't need it. Actually, most of the objects or the specimen that you're supposed to see under the microscope needs staining. Now remove the excess stain, because whenever there are excess also there will be a problem, along with excess water if any, with some soft tissue paper, and then finally prepare it to see under microscope. So these are the what we call the rules related to microscope. So what we're supposed to know is now the do's and don'ts while using the microscope. We'll bring the microscope, keep it, we have to hold it by the base and R. When you put on the stable, you have to just push it a little bit in. And once you put it a little bit in, you want to adjust these objectives onto low power objective. Every time we start with low power objective. It is having the low power objective. And then you try to adjust the light sources before you place any of the slide having the object or the specimen. Once you adjust the light source so that it will go through all the lenses up until the eyepiece, you bring your prepared slide with the specimen or object onto the stage, and you try to hold this slide secured. When you hold the slide secured, it won't fall down. It is adjusted on the low power objectives. Now once you have this adjustment onto the low power objectives, you start to look from the top, opening both eyes with coarse adjustment knob. Once you feel you find the image, you have the right to change middle power objectives. You should take care that their length is different, so you may pull it up and then look down. And when you look down, you may look at, and you may need further magnification. Still you can change your high power objectives. Once you're done and satisfied with the image you got, then you try to draw it if you want to draw it, analyze, record some information. Once that's over, then you move the objective up and then remove the slide. If the slide is permanent, you put it in this box. If it is prepared, then it is going to be washed off. You clean the lenses. And if there are some dirts around here, clean them. And once you're done, you cover it and you put it into the right place, still keeping the right way of holding it. So this is what we mean by the microscope usage and the don'ts and the do's. The next thing that we're supposed to talk about is about cell. Cell is a very unit of living thing. It is the characteristics that is needed, the minimum needed characteristics, so that you can call a matter living matter. And all the characteristics that we've been talking about, like the life characteristics that we've been talking about, they were supposed to exist within the cell. The unique function and structures of almost most of our processes of life is within the cell. So cell is the basic unit of the so-called living world. So it is wise to devote into cell study so that you understand life in depth. Now, what does cell mean? What is a cell? Like earlier, like we said earlier, cell is coined by Robert Hooke when he saw the cell wall of a cork plant, the cork of a plant. And he's credited for that, and he used to be said that he discovered cell. And actually Anton van Leeuwenhoek is the one who saw the moving cells with their cytoplasm, contents of organelles and everything. And he called them, those organisms, animalcules, which now are supposed to be the protozoans, different types of protozoans. Cell study went further by many other scientists, and one of the turning point was done by J. Parkinge in 1839. He was able to find out what protoplasm is. Then that would be followed by M.G. Sheldon in 1804-1881, who lived in this time. He said that cells made plant, or plant made up of cells. And with the same way, Theodor Schwann from 1810 to 1882, he was able to find out that animals are made of cells. And later on in 1855, Rudolf Virchow again come up with the concepts of that cell comes from pre-existing cells. Actually, M.G. Sheldon and Theodor Schwann was not able to know what Rudolf Virchow later on came up. This is what we mean by the fact that M.G. Sheldon and Theodor Schwann were not able to reject the theory of spontaneous generation. They were still believing in it, that cell comes from nothing. But when we come to what Rudolf have said, that will really falsify the theory of spontaneous generation. Now all these people, Robert Hooke, Anton van Leeuwenhoek, J. Parkinge, M.G. Sheldon, Theodor Schwann, and Rudolf Virchow contributed to cell theory. Actually, we consider M.G. Sheldon, Theodor, and Rudolf who contributed to cell theory. And the cell theory, it differs from time to time. The very first cell theory says that cells comes from pre-existing cells. Cell is the unit structure and function of life. Third one is all living matter, or of course, all non-living matter within living things are the product of cellular activity. This was later on modified into the statements that says cell is the smallest basic unit of life organization. Whatever organization in life exists, that's the smallest unit of life organization, an organized life, an organism formation. The second point is cell comes from pre-existing cells. Whenever we have cells, there must be a predecessor of it. Third one is all organisms are made up of one or more cells. There are multicellular organisms. There are organisms that are found in fixed colony. There are organisms found in unicellular. And there are organisms which are multicellular organisms but still not reaching even the tissue stage of organization. So organisms could be one or more in their way of arrangement. So the level of organization is the first statement. The second statement is the origin of the cell, and the third statement is the general organism's organization on Earth. So these are the three theories that are formulated, the three statements of the cell theory that are formulated. So sometimes we come up some sentences that are not applicable to all cells, and a question could be forwarded that could this be considered as a definition of cell, and that will limit if it is defining certain specific groups of cells and not the other groups of cells, or if it is not inclusive statement of all type of cells, then that will cease to be a cell theory. So again, I'll repeat what the cell theories are. The older cell theory says that cell comes from pre-existing cells. Cell is the unit structure and function of an organism or life. And the third one, and different one, rejected one, was non-living matter are produced, or non-living matter exists within living things due to cellular activity, a programmed cellular activity. They don't exist by accident, that means. So those statements are cell theories. Actually, these days, like I said earlier, these three statements are cell theories. Cell is the smallest basic unit of life, organization. Cell comes from pre-existing cells, it's still maintained, and organisms are made up of one or more cells. Cells are classified mainly as prokaryotic cell and eukaryotic cell. The prokaryotic cell, by rule, they are not supposed to have organelles. When we say organelles, these are partitions bounded by membrane found within cytoplasm. Those cell partitions that are found in the cytoplasm, they are not bounded, exceptionally may be called organelle, like that of the centriole, which is a tubular structure that serve as the asters or the spindle fibers that pulls the chromosomes away apart from one another, so that will split those copies of or replicas of chromosome to newly produced each cells from that single cell. So cellular structures may not be organelles or may be organelles. Some cellular structures cannot be organelles, but still they are structures of the cell. So something that the cell owns can be considered as a cellular structure. But to call it an organelle, there must be things that has to be fulfilled, which is the presence of the membrane-bound structures within the cytoplasm. So one of the membrane-bound structures within cytoplasm is the nucleus. And as a matter of fact, having that nucleus is the way that those cell types are named after. Eukaryotic means the true nucleus cell, and prokaryotic means the before nucleus cell or the primitive nucleus cell, which has got no membrane bound. Actually, we don't call it nucleus, rather we call it nucleoid, even if it is running the cell activities like what the nucleus is running the cell activities. So the prokaryotic and the eukaryotic are the major differences of cell, and the prokaryotic type of cells is common among the prokaryotes, especially in the monomer kingdom, and eukaryotic cells are common among the rest of the kingdoms, the Protestant kingdoms, the fungi kingdoms, the plantae kingdoms, and the animalia kingdoms. These kingdoms are having the eukaryotic cell types, and their genetic setup, and their chromosomes, the way their DNA formed, and their numbers and their quantitatively descriptions differ among the prokaryotes and the eukaryotes. The eukaryotes are those cells that have got the organelles, the structures, cellular structures, but there could be possibilities that those cellular structures could be found in some of the prokaryotes, which are not supposed to be found there. And like I said earlier, biology is a science of exception. It comes with lots and lots and lots of exceptions for whatever rule is set. So there could be some prokaryotes that has got an organelle within. The organelle could be temporary organelle, or the organelle may lack some of the normal arrangements that it had in eukaryotes. Like for instance, we have a chloroplast in eukaryotes that run the process of photosynthesis, but when we come to photosynthesis that takes place within the prokaryotes, we can find that this chloroplast is lacking the single, having the single membrane instead of having the double membrane with the stroma and the thylakoid. It's just having the thylakoid without the stroma. So such situation is existence. So when you compare the prokaryotes with the eukaryotes, they are not comparable. and in size and in structures and all of that. But when we want to compare it, if you want to compare, then the structure, the size of the organelle of the eukaryotes could be comparable to the whole lots and stocks of the prokaryotes. So that difference is there. Now, before we go to the differences of the prokaryotes and the eukaryotes, before we go to the differences of the plant cell, animal cell and protozoans, it would be rather good to talk about the cell structures. Actually, when we talk about the cell structures, we have to consider that there are cell organelles and there are cell structures, and one is a superset of the other. Cell structures could be inclusive to organelles, but organelles may have some exclusive structures that are not to be called as organelles. Like, for instance, flagellum. Flagellum is one of the whip-like structures of a cell that most of the time used in locomotion, and they're not the organelle. They are the cellular structure. And again, think about the cell wall. Cell wall is again the product of the cell membrane, but it's still not the cellular structure. So the concepts of organelles and the concepts of cellular structures has to be clear, okay? Has to be clear. The prokaryotes are supposed to have no partitions within them bounded by a membrane, while the eukaryotes are having a number of structures bounded with membrane found within them, and we call them organelles. So organelles are cellular structures, but not any structure that a cell contains. There must be a fulfillment to be done by an organelle so that it would be called an organelle. So organelle has to have a membrane-bound structure. There could be some exceptions like centriole, the tubular structures of the animal cell, but we have to have those bounding membrane within. So this is what we have in terms of the eukaryotes and prokaryotes. As to their names, they're named after not having a full set of nucleus. A nucleus has to have, later on we'll see, it has to have a nuclear membrane with pore called nuclear pore, and within it has to have a genetic material in the form of chromatin condensate, which is the DNA condensed into a chromatin, and another genetic material that takes the information from DNA to a reaction center of cytoplasm and ribosome so that protein will be prepared, and this is what we call the RNA, and it is there in a kind of darkened spots condensed, and we call that darkened spots condensed a nucleus. All this stuff has to have by the nucleus, or the nucleus has to have it. If one is lacking or the whole set is not available, then it's a genetic material, but we cannot call it that genetic material as nucleus. So the naming used this structure as to the types of these cells. That is, in prokaryotes we don't have that nucleus. That means it is the primitive nucleus. While we come to the eukaryotes, eu means true, and karyotes means nuclear material or genetic material. So it has got all that set. So the nucleus is present in eukaryotes, but not in prokaryotes. That does not mean that the prokaryotes lack genetic material. When we talk about those structures in cell types, we would like to refer to different kinds of structural similarities and differences between those types of cells. Within even eukaryotes, you have the plant cell, you have the fungal cell, you have the protozoan cell, you have also the animal cell. And not necessarily that all of them are the same, and not necessarily that all of them are clear-cut, black and white, big differences along the line. So we're going to talk about their comparison in terms of contrast and compare approach. But for now, let us talk about the structures of the cell. Now, if you want to call a thing, a fluid, a cell, or some contents a cell, or a matter a cell, you need to have a cell membrane. It must be a bounding structure to it. And that existence of cell membrane, at least that will tell us something that the formation of cell is there, or somewhat an active cell is there. And the list that cell membrane existence would bring about the existence of cell, like what we have in red blood cell. If you've seen the mature red blood cell, you can see that that mature red blood cell is stuffed with hemoglobin. It's nothing but hemoglobin. But when you come to the naming it, we said red blood and cell. So that red blood cell is termed, now because it has got organelles, a number of organelles, even it doesn't have the list fulfilled structures that you may see in prokaryotes. But that cell has got, though it has got its nucleus earlier, before maturity, it has got the cytoplasmic content and along bordering of the cell membrane. So cell membrane is a very important one. And that cell membrane is normally made up of, like you can see on the picture, the two layers of lipids. When we say lipids, the fats and oils, the molecules of fats and oils that we will see later on. These lipids, which is the molecules of fats and oils, they're in the form of phosphate and then two tails of lipids with a phosphate. We call it phospholipid. So one interface is facing out, having the phospholipid head and the tails inward, and the other is the interface in the in part of the cell from inside, and again it is looking towards the cytoplasmic content. The phosphate is looking towards the cytoplasmic content, and again the tails are inward, and in the middle the two tails are kind of proximal, while the two heads are polarized. Cell membrane naturally has got hydrophobic and hydrophilic area. That means the hydrophobic area are the external and the hydrophilic, the internal part of it, and the hydrophobic area are the internal part are the hydrophilic and hydrophobic, and the external, the interface, the outer part, the heads of the phosphate are the hydrophilic. Naturally, lipids form the cell membrane, and it is with all the control mechanism, because that's the door or the gate in and get out of the cell. So there must be some kind of mechanisms of control, and the mechanisms of control is done by different types of receptors that are made up of carbohydrate and protein, and some of them are protein, and there are channels that allow to come in and come out in an energy consuming or a simple from high to low gradient or concentration movement, and all these things, the selective in and out, okay, the selective in and out, the recognition between cells, the high, okay, they greet each other, cells greet each other by their cell membrane. There are receptors there that are made up of proteins and carbohydrates in a simple sense, and cell membrane in such a way very essential to the cell, because this is the part of the cell that allows it to communicate to the external part, the extracellular stuff. The other thing which is very important is the cytosol. The cytosol is the fluid, the swimming pool that organelles are there in the cell. The cells, the organelles in the cell are not concentrated or settled down or kind of precipitate to a direction or whatever, but they are located at their fixed place, and the viscosity of the cytosol allows those structures to stay in position. Along, of course, with the cytoskeleton, the different cytoskeletons are trans, as you can see in the picture, from a cell membrane to a cell membrane, keeping like a cord, like a rope, keeping organelles there in a place. That's why when we have a picture of a cell, it looks as if they are positioned in such a way that there is a kind of scaffold there, and you put them in, and then they don't move. Actually, they do when necessary. That when they're at a certain division to go, when they're in a process of exocytosis, removing substance out, or endocytosis, getting them in, there you can see some of the organelles moving from one to another place. So you need the cytosol. So we've been talking about cell membrane, we're talking about the cytosol, and the organelles that earlier we talked about the organelles, the structures that are bounded by a membrane found within the cell, and the substances there could be some intracellular structures within the cell that are not organelles. You can say that there are structures called intracellular and structures which are extracellular. And the borderline is the cell membrane. If they are growing out of the cell membrane or from the cell membrane but not into the cytosol, then not into the cytoplasm, then there are structures of the cell but not the intracellular, rather the extracellular. When we talk about those which are into the cytosol or found in the cytosol, then they are the intracellular structures. And out of the intracellular structures, we have the organelles apart from those which are not organelles. So everything is cellular structure, but there are intra, extra, and within intra you have the organelles and non-organelles structure. Actually, the non-organelles are those which are not membrane bound, and mostly they are the characteristic of the prokaryotic cell. The other point we would love to talk about is the cytoplasm, or in a kind of formal way called the protoplasm. The protoplasm is the living organelles, the living substances and the structures found within the cell, along with the fluids inside the cell. And this is what we call the protoplasm. So you can differentiate between cytosol, protoplasm, which is again the cytoplasm, and the cell membrane, the importance of it. So if you want to count cells, you're not going to count the cells in terms of their size. You're going to count them in terms of their having their own cell membrane. If they do have their own cell membrane, then you can count them 1 to 10. And if you've got one big cell with multinucleated, you're going to call it just a big cell with multinucleated, not cells.